Welcome to the 2012 Short Lecture Prize in Life Science and Medicine at HKUST. I'm Mingjie Zhang from Division of Life Science. Today, we are very honored to have two of the greatest scientists in my profession, Professor Arthur Horwich, Professor Franz Ulrich Hartl, to address to us on the year 2012 Short Prize in Life Science and in medicine. The Short Prize is an international award established under the auspices of Sir Run Shaw to honor individuals regardless of their race, nationality, religious belief, who have achieved fundamental breakthroughs in academic and scientific research or applications, whose work has reached in positive and profound impact on humankind. It consists of 3 awards: astronomy, life science and medicine, and mathematical science. Each prize carries a monetary award of $1 million. May I first invite Professor Tony Chang, President of HKUST, to say a few words of welcome.
Good afternoon, everyone. I'm really happy to see the full attendance here, especially I see many young faces here. I assume many of you are students, and it's great that you can turn out to hear great science. There's no substitute for hearing it firsthand, not rather than just reading in a paper and so on. But anyway, I would like to extend my official warmest welcome to all of you today to today's short lecture in life science and medicine by this year's joint winners, Professor Arthur Horwich, Sterling Professor of Genetics and Professor of Pediatrics, Yale University School of Medicine, and investigator of the Howard Hughes Medical Institute, and Professor Franz Ulrich Hartl, Director of Max Planck Institute of Biochemistry in Martinsried, Germany. Together, they won the 2012 Short Prize in Life Science and Medicine for their contribution to the understanding of the molecular mechanism of protein folding. The Short Prize is one of the most prestigious prizes for a researcher. Our 2 distinguished speakers today have won numerous other prizes in addition to the Short Prize. In 2011, they jointly won the Lasker Award in Basic Medical Science. The Lasker Award is often seen as a precursor to the Nobel Prize. Okay. Professor Horwich was elected to the United States National Academy of Sciences in 2003, and Professor Hartl was elected as a foreign associate of the United States National Academy of Sciences in 2011. Proper protein folding is essential for many cellular functions, as misfolded proteins can be toxic to cells and are involved in many degenerative diseases. The work of the 2 laureates has helped to explain what goes wrong in diseases of protein folding, which will help in developing new therapeutic approaches to these kinds of diseases, such as amyotrophic lateral sclerosis, ALS, I hope I got that right, Parkinson's, and Huntington's disease. Before yielding the floor to our distinguished speakers, I would like to thank the Shaw Prize and Sir Run Shaw, who established this prestigious international award for bringing the distinguished Shaw lecturers to our university every year. The opportunity to be enlightened firsthand by brilliant minds is one we should all cherish. Lectures by these 2 great scientists can certainly inspire all of us, especially our younger audience here today, to set higher goals and achieve greater things. HKUST has a very strong team in life sciences, including 2 members of the Chinese Academy of Science, one of whom is here today. The Division of Life Science, like the rest of HKUST's academic departments, is doing cutting-edge, innovative research and nurturing young talents. The visits by Professor Horwich and Professor Hartl will certainly provide a fertile opportunity for our colleagues to interact with the world's foremost scientists. We look forward to more collaboration in the future. For now, let's sit back and watch two great minds at work. Ladies and gentlemen, it is my honor to present two great life scientists, Professor Arthur Horwich and Professor Franz Ulrich Hartl.
Thank you, Professor Chen. Before we start the lecture, can I invite Professor Chen to present a small token of souvenir to Professor Horwich and Professor Hartl. First to Professor Horwich. So the souvenir is actually a replica of the mural on our sundial, represents ancient Chinese science and technology innovations. Okay, now can I invite Professor Hartl? Professor Hartl, can you stay? Professor Chen, can you stay? Can you stay? Professor Horwich and Professor Hartl, can you, three of you together? Yeah. Yeah. Thank you. Thank you. Now, may I invite Professor Arthur Horwich to deliver his lecture. The title is Chaperone-Mediated Protein Folding.
Okay, should be okay. 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 Well, President Chen, Professor Zhang, Stephen Kong, and all of you, thank you so much for having us here today. We've had a chance to tour your absolutely exquisite, beautiful campus and meet with a few colleagues already, and it's been a wonderful day. I'm really honored to be able to visit HKUST. And of course, we're hugely honored to be recognized with the Shaw Prize. The ceremony last night was really like a dream certainly for me. I still have to pinch myself to imagine that I was recognized in this way. And I think, as Ulrich has commented several times, and I think it's very accurate, it's really on beyond to be honored as a scientist when really you're getting to just have fun every day in your laboratory tinkering with biological molecules and trying to understand Mother Nature. So it's been so special. So I'd like to essentially tell a story that focuses on the chaperoning machine. Ulrich will carry that story further to additional machines, as well as saying more about the chaperoning system. But I thought I would just sort of give you a sort of historic view of how things happened, so that particularly for students in the audience, you can see that things don't just automatically emerge in the literature, that groups of people think about problems together and devise experiments to try to understand findings that they have. And often we sit there and doubt our findings and wonder whether we're right, and just continue to do experiments until we're convinced about something. So, of course, this story concerns the general issue of how information is transferred in living cells. So, of course, it's encoded in DNA, and it's transcribed into RNA, And we know that then at the ribosome, there's translation of polypeptide chains as strings of amino acids, with each amino acid one of 20 different possibilities. And that the proteins that effectively carry out the functions inside of us, enzymes, cytoskeletal proteins, hormones, hemoglobin, channels, receptors, and so on, that these proteins each have characteristic amino acid sequences that ultimately dictate a folded form that is characteristic for each amino acid sequence. And that folded form is the active form of the protein. And so we're really focused then on this final step of information transfer, that of folding of the initially made string of amino acids to a functional structure. So the granddaddy experiment in this field, a spectacular experiment that I heard about when Christian Anfinsen received a Nobel Prize in 1972 for this experiment. I was an undergraduate at Brown University at that point. I remember picking up the newspaper and just being stunned by this experiment, was a test in which Anfinsen asked whether you could take a folded protein, in this case ribonuclease, which has a few disulfide bonds shown here in color. He broke those disulfide bonds with mercaptoethanol and unfolded the protein using 8 molar urea as a denaturant. And this random coil protein was then asked whether it could find its way back to the native state spontaneously in a test tube, whether the purified, unfolded protein could spontaneously refold. And astonishingly, it was able to do so, and fairly efficiently, ultimately, using dialysis to remove the urea and then air oxidation of these sulfhydryl groups. So that implied that the amino acid sequence of a protein contains all of the information to allow it to fold into its unique native state, which he presumed to lie at an energetic minimum. So just an absolutely astonishing result. And that was a result that pretty much carried people's thinking about how protein folding occurs for several decades. But there were some stumbling blocks. So for example, many proteins that people tried in these kinds of experiments were not able to refold spontaneously in vitro. And the result was a misfolded, aggregated collective of protein molecules. If you spun the sample tube, you saw white precipitate at the bottom of the tube, indicating that your protein had misfolded. And of course, there was no biological activity recovered. In addition, as biotechnology sort of got born, it was possible to express human proteins, for example, in E. coli cells to try to produce insulin, growth hormone, and a variety of other proteins, interferon, and what have you, in the early days. And the results were very disappointing in many cases, where the protein was overexpressed in E. coli and produced misfolded, aggregated, so-called inclusion bodies that localize at the terminal ends of the bacterial cell. And so there had to be something more going on. And the conclusion that was beginning to be reached was that for many proteins and under cellular conditions, folding is kinetically difficult. Anfinsen's principle was correct, that the primary sequence directs folding to an energetic minimum that's the native state. But in living systems and under some conditions, the chain could go wrong. That is, an error could occur during folding, for example, due to physiologic temperature, which was relatively high as compared to the room temperature test tube experiments of the late 50s, early 60s. And in cells, the local concentration of solute is very high. For example, the local concentration of cellular proteins could be 200 to 300 mg per mil, so it's almost like a gel-like situation in which polypeptide chains are trying to refold. And so the situation could be described by these so-called energy landscapes. You could think of the ribonuclease experiment that Anfinsen carried out as a straight shot of a lot of different conformations, the ability of any of them to quickly find their way to the native state by going down an energetic landscape and reaching the native minimum. But under cellular conditions, or with proteins that are more complex, that have multiple domains, for example, The polypeptide could start on parts of the energy landscape where it would run immediately into a local minimum and where it would be stuck. And particularly in a physiologic situation where you have to be able to make a functional protein on the time scale of seconds or a minute or two, this could be damaging to the cell. Because number one, you fail to be able to achieve a functional end product protein that the cell needs in some sort of urgent fashion. And number two, these kinds of misfolded states are capable of coalescing with each other to form aggregates that are harmful to cells and harmful, as we know, in settings, for example, of neurodegenerative disease. So again, in the historic perspective, as we came toward our initial experiments, there had already been a study done by Hugh Pelham's group that was a sort of a connector experiment. Pelham was looking at actually heat-shocked tissue culture cells and noticed that if he overproduced a particular protein called heat-shock protein 70 in the tissue culture cells, the damage that he saw inside those cells, particularly to the nucleolar compartment, was reduced. So somehow the overexpression of this heat shock protein, and I should say that heat shock proteins had been discovered a long time before these experiments of the early 80s were carried out, but their functions had been completely unknown. They had been thought to have to do with glucose metabolism or stabilizing nucleic acids. There had been all sorts of speculations about what they did. But here Pelham was showing that a heat shock protein could actually act through some additional experiments he carried out with isolated nucleoli, could act as a sort of a molecular crowbar or chaperone, preventing or reversing illicit, unwanted interactions between protein molecules, preventing them from aggregating. And in 1986, this model of his was published in a sort of a news and views context in Cell, where he suggested that ATP could bind to HSP70, and then the action of its hydrolysis allowed HSP70 to separate aggregating proteins from each other. The final sort of understanding of how HSP70 works is a little bit different than this, but the principle that heat shock proteins have something to do with preventing aggregation was certainly brought to the fore by the early work of Pelham. The question that really came to us, though, is not so much to deal with stress conditions, but normal conditions. Is there assistance by cellular components to de novo protein folding under normal conditions? So now we're talking about under the normal conditions where a cell is at its normal temperature and just undergoing normal metabolism. Is there a need for any kind of assistance to polypeptide chain folding? Okay, so I need to set this up. It'll take me just a part of a minute. But we, at that point, were studying how proteins are imported into mitochondria. So historically, as a physician working on a severe metabolic disease that involved a mitochondrial protein, a protein of the urea cycle of the liver called OTC, we were interested in understanding how OTC actually gets into mitochondria. We knew that the protein, because it was nuclear encoded on the X chromosome, was being translated in the cytosol as a larger precursor protein with an N-terminal targeting peptide that says, take me to mitochondria. It's essentially a ticket that directs the protein to mitochondria. You could attach other proteins to it and it would take them to mitochondria as well. We were interested in the machinery of uptake into mitochondria and had moved from a mammalian system into a yeast system. So the advantage to yeast is that they're haploid, and as you know, you can do genetics a lot more easily in that kind of a system. And we had already shown that you could express human OTC in yeast, and even though it had never seen it, its mitochondria efficiently took it up and made the active homotrimeric enzyme in the mitochondrial matrix. There is an OTC enzyme in yeast, but we knocked it out without any ill effects on the cell. It's actually a cytosolic enzyme in yeast. So, to bring you to the point, there had been an experiment carried out and published in 1986 from Gottfried Schatz's group in Switzerland that suggested that for newly made mitochondrial proteins to get into mitochondria, they had to occupy an unfolded conformation. So if you took, for example, dihydrofolate reductase and let it completely fold and stabilize it by adding the ligand methotrexate, even with a signal peptide attached, it would not go into mitochondria. But if you now unfolded the protein, took away that ligand, took away the methotrexate, now the protein would readily go into the mitochondria and reach an active form. So the question that we raised as we started to carry out a screen in Baker's yeast was, after a protein enters the mitochondrial matrix space, does it spontaneously refold to its active form the way you might predict from the Anfinson observations, or does it need some sort of assistance in order to reach the native state? Well, here we had a bank of yeast mutants that we made in which we assessed first for viability, because we knew that if you can't make new mitochondria, you have an inviable cell. So we essentially used temperature-sensitive lethal mutants. So the cells could grow at 23 degrees, but when you shifted to 37 degrees, they arrested their growth. So we started with that bank of TS lethals, and now we inquired out of each mutant individually, if we turn on expression of OTC after shift to the non-permissive temperature to 37 degrees, could you make OTC enzymatic activity? And so the particular kind of mutant we were looking for was a mutant in which you would not make any OTC enzymatic activity, for example, due to mutation. However, the protein would get into the mitochondrial matrix space. So the import process would occur, but then at the end step, there would be no correct folding, no enzymatic activity, and we would see the mature sized protein with its signal peptide cut off. And so the idea for looking for this occurred to us late one night, and literally within a week, Ming Cheng, one of my early graduate students who is a young physician from Taipei, observed that in fact we saw exactly that phenotype with a mutant that she called MIF4, or mitochondrial import function 4. Actually, it went by the handle alpha 143 to start with, because it was the 143rd mutant in a huge collection. And that observation stunned us, because suddenly we thought, this is heresy. We're challenging something that nobody's going to believe. I mean, how are we ever going to convince anybody that there's such a thing as a folding assistant or a folding machine inside the mitochondrial matrix? And so what we did as a second experiment was to ask, well, what about an endogenous yeast protein, the F1 beta ATPase subunit? Could it be affected also in this particular mutant? And so Ming carried out an extraction procedure that showed that, in fact, F1 beta also was affected. We failed to find the subunit in this normal stalk structure where the F1 ATPase points into the matrix space. And so F1 beta appeared also to be affected. It got its signal peptide was cleaved, but it was biologically inactive. So that further bolstered the idea that even endogenous proteins coming into the mitochondrial matrix might be affected. But now we were stuck, because we really weren't sure how to proceed to characterize something that we thought was so heretical. And we were very lucky at this point, because at this point, Ulrich and Walter Neupert called and said, we understand you're studying some mitochondrial import mutants. Would you like to join hands with us a little bit? We're pretty good at some of the biochemistry that we suspect you guys might need some help with, which is an understatement. We were, 4 people in the lab in total at that point, and we hadn't done a lot of mitochondrial biochemistry from yeast. And so it was a delight to go visit them the next week and present some of our data. At the end of the talk, I decided to present some of the data from this MIF4 mutant, and I think they were pretty surprised. They were concerned that we might be missing something, that proteins might actually be jamming in these import sites, you could have a protein jammed in such a way that its signal peptide would be cleaved here on the matrix side, but the rest of the protein would be jammed in an import channel, and therefore it would be unable to fold to the active form. So that would be a different interpretation of the kind of result that we were thinking about, namely that there was some sort of active moiety in the mitochondrial matrix that was assisting folding. So Ulrich suggested that what we should do is really isolate mitochondria from the mutant strain and ask specifically whether proteins had reached the matrix compartment by adding exogenous protease. So if the protein was dangling out with part of it in the cytosol, the protease would cleave the protein. If, on the other hand, the protein was fully imported into the matrix space, it would be resistant to exogenously added protease. And about 2 weeks later, he called and said, "Oh, really great result. The protein seemed to be going completely into the matrix space." So this led us to further believe that we were looking at something that had to do with folding of these, with the refolding of these unfolded proteins as they came through the mitochondrial membranes. And then together, and with really major help from them, we were able to study a number of other components of the mitochondrial matrix and show that they also seem to be failing to undergo normal folding and biogenesis. One protein that we were really excited about was the iron-sulfur protein, because that protein, in contrast to some of these others that were all oligomers, that leave open the possibility that maybe we were looking at a defect of protein assembly, not protein folding, this protein is a monomer during its lifetime in the mitochondrial matrix. It has to undergo 2 cleavages before it ultimately lodges in the inner mitochondrial membrane. And we found its cleavage to be profoundly disturbed, implying that the protein had misfolded as a monomer and had failed to really, it was thus failing to reach its native form in that lodges in the inner mitochondrial membrane. But in addition then, in parallel experiments, Ulrich with Joachim Ostermann in the lab used a obligate monomer, soluble monomer, dihydrofolate reductase, and sent that into mitochondria and showed that indeed that protein, when it's newly imported in the deficiency of ATP, it becomes associated with a large molecule and occupies an unfolded conformation while associated with it. If you then added ATP back to that system, you could watch DHFR leave that large complex and assume a native-like conformation. So these kinds of bits of data were crucial to our feeling that we were studying a protein folding machine. So now, to cut a long story short, we ultimately rescued the mutant with the yeast library, and the protein that came up was called heat shock protein 60. And that was actually a name that we all collectively gave to it. It was a protein that had been actually identified a year beforehand as a modestly heat-inducible protein in the mitochondrial matrix by Richard Hallberg working in Iowa using T. thermophila. But we knew that he was working on the yeast version of this, and we called him up on a Saturday afternoon in the summer of, I believe it was probably 1988, and said, "Do you have any sequence data on this protein?" Because he was cloning it using a Lambda GT library. And here we had a complete gene by rescuing our mutant. And Ming Cheng and Richard Hallberg sat there and did a dead match of the nucleotide sequence of HSP60, what we collectively came to call HSP60. Now, Hallberg already knew that this molecule that he had been studying, this mildly heat-inducible protein, was a double ring assembly. So now we could impute the protein folding function in the mitochondrial matrix space to a double ring assembly that was known also to look something like a bacterial protein called GRO-EL. So this started to look like there was some generality to what we were studying. Notably, HSP60, however, even though it's a heat-inducible protein, is essential under all conditions. Both our group and Hallberg's group carried out gene deletion experiments in yeast, and at all temperatures, this was completely lethal to the cell. So this suggested that this folding function is crucial for biogenesis of proteins in the mitochondrial matrix under all conditions. Finally, I guess I should comment that HSP60 is required for its own biogenesis, as Ming Cheng showed shortly thereafter. If you tried to import HSP60 subunits of a wild-type nature into an HSP60 deficient strain, you couldn't make any new complexes. So the machine actually assists the folding of its own subunits, at least in the case of mitochondria. So indeed, the generality of chaperonins as folding machines came strongly through to us as we compared sequences of the machine that we were looking at with the GRO-EL protein in bacteria and with a protein that John Ellis and co-workers had imputed in chloroplast assembly called the Rubisco binding protein inside chloroplasts. So these proteins bore close to 60% amino acid identity in their sequences right across. And then shortly after that, Jonathan Trent, a postdoc in my group, but working both with us and with Ulrich, made a further connection of a thermophilic factor from archaebacteria that was a double ring assembly that could confer thermotolerance to archaebacteria as being a second limb of chaperonins. When we sequenced this particular subunit, we found that it was closely related to that of a subunit of another assembly that had been in search of a function called the TCP1 complex or TRIC complex, which is the chaperonin of the eukaryotic cytosol, crucial for folding actin, tubulin, beta propeller proteins, and a host of other components. This, however, in contrast to many of these other chaperonins, is not really a heat shock protein, but is essential for life. Interestingly, it has 8 different subunits per ring, whereas these tend to be homoologomers. And whereas this family has detachable lid structures, which I'm now going to come to in terms of the folding mechanism, these components have a built-in type of lid structure. So this is us in the early days. You know, we didn't have email or Skype in that era. And so Ulrich and I literally went back and forth across the Atlantic quite a few times to be together to discuss mechanism and how things might be working. Here's Manojit reading to us, maybe telling us how the story might turn out in the end. But we had some wonderful times together. My wife calls our late night discussions grow speak, short for GRO-EL conversation. And we had a lot of grow speak during those days. So what happened next was a reconstitution experiment in which George Lorimer's group, and then followed by Jörg Martin and Ulrich and his group, were able to reconstitute folding using purified GRO-EL. Again, this double ring assembly, 7 subunits per ring, a tetradecamer, and its cooperating component that shares an operon with it called GRO-ES, a single ring of 7 subunits, each of about 10,000 molecular size. And what was observed was that in the presence of GRO-EL, a protein like Rubisco, diluted from denaturant, could be prevented from quantitatively misfolding and aggregating, and would form a 1-to-1 stoichiometric binary complex. So Ulrich and Wolfgang Baumeister could observe this in negative stain EM kinds of analyses, and our own group observed this using DHFR as a substrate and gold labeling the DHFR, and then using scanning transmission EM with Joe Wall and Jim Hainfeld at Brookhaven National Lab. We could actually see the gold particle in the central hole of the ring assembly, and we could also see that that gold particle tended to be localized near the end of the cylinder, which corresponds to the peptide binding apical domains, as you'll see in the next slide or two. In a second part of the reconstitution reaction, one could add GRO-ES and ATP, and then something happened, magic happened over a period of a few minutes, and the native protein was formed. GRO-ES was released from GRO-EL, and in our early experiments, we could detect that it seemed that GRO-ES had to be associated with GRO-EL in order for productive folding to occur. We had no idea what the topology of that would be, but it seemed to somehow couple the folding reaction to the assembly. So you get a flavor for the idea that we had inklings about things ahead of really understanding them, but we just kept at it, and eventually we got to a molecular understanding. And in our case, the really major step toward molecular understanding came in the form of a collaboration with the late Paul Sigler, who was really a wonderful X-ray crystallographer and teacher. So Paul had just come to Yale from the University of Chicago This is him and his group looking at the trip repressor operator structure that had just been published in Nature in 1988. And this is Andrzej Ohimiak in the group with whom we worked really closely to ramp up production of GrowEL in order to generate crystals. And the mastermind of ultimately solving the structure was Zbyszek Awinowski. So Awinowski was an astrophysics graduate student who kept walking into the Sigler lab as a glass washing person to just make a few dollars, but kept looking over the shoulders of people and solving their X-ray structures. And eventually Sigler convinced Awinowski that he should really work on X-ray crystallography, and he joined the lab. And for any of you who do crystallography, you know that all the data reduction programs come from Zbyszek. So it was wonderful to have him involved. I should just say that the way I originally met Paul was I got sent across town where he was located. I was down in the medical school to discuss an administrative matter with him. And after about 30 seconds, he said, okay, we've solved the administrative matter. Let's talk about these ring machines that you guys are working on. I'm really excited about the symmetries that you have. And you know, you're never really going to understand how they work unless you crystallize them. And I said, well, I completely agree, Paul. Let's get going on it. And that was it. That was the start of the collaboration. So Paul brought Zbyszek into the room and said, well, okay, can we handle this computationally? And he said, absolutely, we cannot. This molecule's too big to handle at this point. So this is 1990 or so. But he said, it's going to take you 3 or 4 years to generate decent crystals of this molecule. And by the time you do that, the programs will be there, computing will be better, and you'll be able to work your way through this. So that's exactly what we did, was to set out to generate decent crystals. And that was exactly right. It took us 3 years to generate good crystals. And they came in the form of crystals that my student Kirsten Bragg, who came to visit from Berlin for 6 months but stayed for about 5 years, generated. There were these beautiful orthorhombic crystals. So basically they came from what was a long line of molecules that we tested that actually turned out to be a variant of wild-type GrowEL that was an accident of a PCR reaction that just substituted 2 amino acids harmlessly. Actually, the molecule is fully functional. But those 2 amino acid changes enabled her to produce these beautiful crystals. So she had a wall of trays that's about like this, from which she found a tray in which these crystals sat in one of the hanging drops. So it was a huge undertaking with a lot of different molecules, massively overproduced. I should say that by the time we could make these crystals, Andrzej and I had figured out how to overproduce GrowEL to the level of about 98% of total E. coli protein using a sort of trick promoter vector system that we set up. And so you'd see just a single band on a gel after overexpressing GrowEL, and you just had one step, and then you were setting up trays. So that really made a huge difference. So I won't go through all the structural solution, but we'll just say that it was wonderfully exciting to collect native data, and then for all of us to learn how to make heavy atom derivatives, because that was the only way you could do this. Even a selenomet would have been pretty tough to work with at that point in history, or even now. But a single ethyl mercury derivative allowed us to solve the phase problem. And Zbyszek, as true to form, solved the structure in a single day once we got him the heavy atom data. And what we saw basically was what had been suggested from EM, 2 back-to-back rings. So the equatorial domains of each of the subunits in these rings, and again I should say that each subunit is made of 3 domains. The equatorial domains essentially are a platform that hold this assembly together. So they make contacts going around the ring and across the interface between the 2 rings, tight contacts that essentially hold the assembly together. And ATP binds on the inside aspect of these subunits. And so this is the sort of generator for making the other domains move. And they make rigid body movements. So these are the intermediate domains that are hinge-like structures. So you can picture a hinge at the bottom, at the top of each of these intermediate domains. And here's the business end of the machine, the apical domains that lie at the terminal end of the cylinder. And so they're basically pretty much flying in the breeze, just making little contacts with each other that are quite weak. But they have a hydrophobic binding surface on their inside aspect. And that was really the key to sort of understanding what's going on here. So if you were standing inside of the top ring of GRO-EL and looking at two subunits on the back of the ring, this is what you'd see at the level of the apical domains. So there are a tier of 3 secondary structures, a helix, and an underlying segment. And on each of these structures are hydrophobic side chains pointing directly into the solvent. And so that was the aha. When we mutated any of these, GRO-EL was unable to bind any polypeptide. And so any of them individually abolished polypeptide binding. So that suggested to us that indeed the way these machines work is when a protein is in its native state, hydrophobic surfaces are buried to its interior. When the protein is unfolded or in a misfolded state, it's exposing hydrophobic surfaces, and they are essentially sticky surfaces that are captured on this hydrophobic binding surface. So there are really 7 of these going around a ring, and that binds the non-native polypeptide and prevents it from aggregating with some other polypeptide inside the cell. And so the multivalency of that binding was shown first using genetic experiments where we could make GRO-EL rings as single molecules at the coding sequence level and then mutate the apical domains in any combination or permutation. What we found was that you need 3 or 4 consecutive good functional apical domains in order to bind a non-native polypeptide. But then more recently, in collaborative studies with Helen Sable's group at Birkbeck College in London, we've observed that a non-native protein, while bound to GRO-EL, indeed associates with 3 or 4 contiguous apical domains. So here you see the non-native state of malate dehydrogenase while associated with an open GRO-EL ring. And so this multivalency of contact, once again, assures efficient binding of a polypeptide and may be able, in effect, to exert some force to pull it apart. If it's kinetically trapped, there might be some pulling force by virtue of multivalency experiments that are supported by fluorescence experiments from Hayes-Rye's group and also from Ulrich's group. So again, the general action not only of the chaperonins but of all molecular chaperones appears to be to deal with greasy surfaces, with hydrophobic cores that become exposed when a protein is subjected to conditions either during folding or during stress where these surfaces become exposed. And so in order to prevent aggregation, the chaperone proffers its own hydrophobic surface to mask those exposed hydrophobic surfaces of a non-native protein. So in the case of the chaperonins, the geometry is a collapsed protein inside of a cavity being physically associated with. In the case of the HSP70 proteins, as you may hear some more about, it's a different geometry where the HSP70 protein has a little hydrophobic arch that binds to a short extended segment of polypeptide that exposes hydrophobic side chains. So the geometry of binding by the different chaperones is different, and hence there are different involvements in pathways of protecting the cell by binding non-native proteins. Okay, so now I want to come to the folding aspect of the chaperonin reaction. So where does folding occur? When we took unliganded GRO-EL structure and superposed one of our favorite substrates, rhodonese, on it, we saw a terrible steric clash with the apical domains. It seemed impossible that folding could occur in the machine if you had things bumping against the walls like this. But our collaborator, Helen Sable, changed our thinking in a major way. So Helen has been our EM collaborator since 1992. Here she is sitting on in her garden in London on a day when it's not raining and everything is in bloom, amazingly. And so what she observed was that when GRO-ES binds to GRO-EL, the apical domains of the bound ring open up and there's now a cavity space of substantial size. Here's the opposite ring, which is not GRO-ES bound. I should say that in most nucleotide conditions, GRO-ES binds asymmetrically, and the whole ring assembly functions inherently asymmetrically. So there's cooperative, I should say, GRO-ES binding requires ATP, to be present in a ring. ATP binds cooperatively within rings and anti-cooperatively between them. So here's the control ring where you see little or no open central cavity, whereas here you have a large cavity that's available. So Jonathan Weissman, who was a postdoc in the group at that point, carried out an order of addition proteolysis experiment to ask whether a polypeptide substrate could in fact be housed underneath the GRO-ES lid inside of a GRO-EL ring. So he carried out 2 orders of addition. In the first order, he added GRO-ES first to a GRO-EL ring, encapsulating one of the rings, and then added polypeptide, and then added exogenous protease. And when he did that, he completely digested the protein. There was no protein found to be protected from protease. But when he did the opposite order of addition, adding polypeptide first, followed by GRO-ES, now with polypeptide bound, GRO-ES could come on randomly either to this ring or to this ring. When it came on to this ring, it was accessible to protease. When it came on to this ring in this fashion, it was completely resistant to proteolysis, and he carried out a variety of topology experiments that proved beyond any doubt that a polypeptide housed underneath GRO-ES was protected from exogenously added protease. So you could have what we call a cis-ternary complex where a polypeptide was housed underneath GRO-ES in a GRO-EL ring. Well, did that have anything to do with productive folding? So Wayne Fenton, who's been with my group for about 30 years, formed either trans complexes or cis complexes, and I won't go through the details of their formation, but then discharged them under a single turnover experiment with ATP and asked which of these could produce OTC enzymatic activity. And what he observed was that cis complexes were productive and trans complexes were not productive. So indeed, this topology of a polypeptide underneath GRO-ES inside a GRO-EL ring could at least commence productive folding at that point. The question was whether folding could go all the way to completion in a so-called cis chamber in one of these GRO-ES encapsulated GRO-EL rings. And so the problem is that under normal conditions in the presence of ATP, the GRO-ES lid binds and releases from GRO-EL about every 10 seconds. And so you can't really make a stable folding chamber under those conditions unless you trick the system somehow. So the trick that we used was essentially to stably lock GRO-ES onto GRO-EL by making a single ring version of GRO-EL, because it turns out from work of George Lorimer that the action of ATP in the opposite GRO-EL ring, when it's present, ejects GRO-ES from the so-called cis ring. So if you took away this ring altogether, now there was no ejection signal at all available. And so when GRO-ES bound to this ring, this single ring version of GRO-EL, it was stably locked on. So now imagine an order of addition where you take the single ring version of GRO-EL, SR1, add a polypeptide to it while the ring is still open and able to accept a non-native polypeptide, then add GRO-ES and nucleotide, and now ask whether the protein could reach the native state inside of this stable chamber. And I should say at the same time we were doing these experiments, and completely unbeknownst to us, Horwich's group with Mark Mayhew was doing the same kind of experiment with dihydrofolate reductase and coming to the same result, namely that protein folding to the native state could occur all the way to the native state in this cis chamber. So here's the data. Here's Rodinese activity recovery during a folding reaction, either at wild-type GRO-EL or at this stable SR1 GRO-ES ATP complex. And here's that complex, the SR1 complex, actually gel filtered at each time point, taking the 400,000 Dalton complex and asking whether it has any enzymatic activity. And indeed, the recovery of activity is identical to what we observed here. So this indicated that Rodinese could completely fold to the native state inside of this cis folding chamber. Well, what does a cis folding chamber look like? Well, to get at that, we went back to X-ray crystallography, and once again, several years were required. Here, the major working force was Xiaowei Zhu, a postdoc from Paul's lab, who figured out how to freeze GRO-EL, GRO-ES crystals. That was something that none of us were able to do for quite a long time. And Xiaowei figured out a swish procedure through a sugar and then freezing in the liquid nitrogen stream and then directly collecting diffraction data at the synchrotron. And what he was able to do essentially was do a molecular replacement solution, but ultimately it was a much finer study than that because he ultimately threw out all the phases and just used amplitudes and 7-fold non-crystallographic symmetry averaging so that there would be absolutely no bias to this structure, and came up with this end-state structure. And what you see is GRO-ES bound on top of a GRO-EL ring. The ring to which it's bound has undergone large conformational changes, and the opposite ring, the so-called trans ring, looks just like a GRO-EL unliganded ring, as if nothing had happened to that ring. And so here's what this looks like as a C-alpha trace. So here's unliganded GRO-EL, 45 angstrom diameter cavity with all that hydrophobicity on the wall here. And here is a GRO-ES bound ring, 65 to 70 angstrom diameter across here, and also vertical height. Here's GRO-ES encapsulating this ring. And you can imagine a polypeptide chain folding in this chamber in complete isolation. And I think that's really the trick to the machine, is that folding occurs here in solitary confinement, where a polypeptide does not have a chance to aggregate. It does not have a chance to form a physical association with anything else. And we think that the walls of this chamber are probably relatively passive to the folding reaction. That's not absolutely shown, and there's many experiments that all of us are doing. But we would suggest that Anfinson's principles are basically at work with the primary structure of the chain guiding it to the native state. And again, here is a description of the rigid body motions that set up the folding chamber. The GRO-EL subunit of an unliganded ring undergoes large rigid body movements. In particular, the apical domains undergo a 60-degree elevation and a 90-degree clockwise twist. So all this initial hydrophobic surface used to capture a non-native polypeptide now points out of the board at us, and in fact is making contacts between neighboring apical domains. And there's a new surface now lining the central cavity, and this now is a hydrophilic surface shown by all of this blue. It's actually quite electrostatic, a little bit of net negative charge, but we think this is sort of a no-stick surface that keeps the polypeptide, even though it's probably having translational and rotational collisions with the wall, it keeps it essentially in a situation where it can undergo folding using the Anfinson principles. And so this essentially, this image captures GRO-EL both in its binding proficient state, where it exposes hydrophobic surface of an open ring that can capture a non-native polypeptide, and shows it in its folding active state in the cis ring that's GRO-ES encapsulated, where you've replaced all this surface with hydrophilic surface that favors production of the native state. So just one comment about the role of ATP in this machine, and then I'm going to close off. It's ATP binding in this machine and not hydrolysis that triggers productive folding. So this is an experiment that Hayes Rye carried out with what we think is the really minimal folding active state of GRO-EL, a single ring version that's been mutated in such a way that ATP can't hydrolyze very efficiently. Simply binding ATP in GRO-ES allowed Rubisco, in this case, to refold in a stable chamber here to the native state. So the energy of ATP and GRO-ES binding is enough to eject the polypeptide off the cavity walls as those apical domains open and move out, and deposits it basically in this folding chamber to try to reach the native state. And in fact, the gamma phosphate is crucial for this step because we could take a preformed ADP GRO-ES complex with polypeptide in it, where polypeptide is actually stuck on the wall as judged by fluorescence anisotropy experiments. And despite that this is an encapsulated chamber where exogenous proteases won't degrade the rhodonese that's used in this experiment, if you now add either a transition state analog or beryllium fluoride, which is a ground state analog, to simulate the gamma phosphate, everything breaks loose here, the polypeptide comes off the wall, and it now folds at a rate that's just like a wild type reaction. And so the gamma phosphate of ATP is being used to provide essentially 40 kilocalories per mole of rings of energy in binding that somehow jiggles the machine and shakes the polypeptide off the cavity wall so that it's now productively folded in the cis chamber. So in summary then, thinking about the entirety of the reaction cycle with respect to nucleotide, again, as I said, ATP binds cooperatively within rings and anti-cooperatively between them, such that one ring will be folding active and the other will be essentially unoccupied at any given time. The folding active state of a ring is the longest state of the reaction cycle, about 10 seconds in a room temperature reaction. And then this is followed by a hydrolysis event. And it's a hydrolysis event that is really not governed by what polypeptide is present or whether there's polypeptide at all. The machine can cycle without a polypeptide being around. Once ATP hydrolysis occurs here, it gates the entry of ATP and of non-native polypeptide into the opposite ring. As we've recently measured, ATP arrives at least 10-fold faster than non-native polypeptide, but the real slowpoke to get to the ring, fortunately, is GRO-ES. So that really orders the addition of these ligands in such a way that you now make a new folding active ring on what had been the inactive ring by an order of addition. So now I want to close by just showing you a movie that just sort of connects all the dots of this reaction. So it's both the crystal structures and a lot of EM work done with Helen over the years. And so we're just basically connecting the dots of the various states. So you just saw a subunit colored for its domain structure. Now you're looking down the barrel of the machine. Here's all that hydrophobic binding surface at the apical level. I should say the rings are blocked at the equatorial level by C-termini that are disordered, so it's really two separate folding chambers. ATP gets there first to an open ring, and it causes a little bit of elevation and counterclockwise twist. But that favors polypeptide binding, which again is slower than ATP binding. And then again, the slowpoke, GRO-ES, comes through its own hydrophobic surfaces and associates with the exposed apical surfaces. There's this huge rigid body movement that I explained a few slides ago, and now you see folding, whatever that means. Well, again, in our opinion, this is a passive process. At the end of 10 seconds, hydrolysis occurs, free phosphate leaves, ADP will leave, but that gates the entry of ATP into the opposite ring, and polypeptide will bind following these movements of the apical domains. Meanwhile, on the cis ring, polypeptide and GRO-ES left, out comes ADP, and now this ring is folding active. And so that's the slow motion of this. Now I want to show you the reaction as it would occur in real time. This enforces the idea that the transitions are very fast. So this ring is now folding active, and now this ring is going to become folding active by virtue of ejection, arrival of hydrolysis here, ejection of ligands by ATP binding here. And you can see that all of that occurs in the space of about 1 second. So the machine spends most of its time in the folding active state, which is obviously desirable for cellular metabolism. So I want to just close by acknowledging the wonderful collaborators I've had, and these are really the prime moving people. Ming Cheng, my student who isolated the original HSP60 mutant, Horwich, with whom we have had just wonderful times over the years sorting out how this thing works. Kirsten, who pulled the crystals, Spishek, who solved the structure, Xiaowei, who is the hero of the GRO-ES ADP structure. Helen's been our longtime collaborator. I should say we've recently published with her concerning some further detail about how ATP actually works in producing that elevation and counterclockwise twist. And Paul, who passed away in the year 2000, we dearly miss him. There are probably a myriad of additional experiments that we could have carried out related to substrate binding that I still can't quite figure out how to do that would have been possible if he were alive, I think. And on the mechanism work, Wayne Fenton, as I mentioned, who's been with me for a long time. George Farr, who came for a couple-year postdoc and stayed for 15 years. Christina Furtak, who's been with me making mutants since the very first day I was a junior faculty member. Hayes carried out all the fluorescence experiments that really watched the machine move in real time. And Jonathan, who sorted out a lot of the topology experiments in the early days. And actually, there's been a whole host of other people who have contributed that I won't even go into, but all of them brought something to our understanding of this machine, and all of them were really fun to interact with. And I guess that's really what it's been all about for the 20 years we've been working on this, is just really having fun trying to understand how this beautiful machine that Mother Nature came up with works. So thanks very much for your attention. Thank you again for the invitation.
Thank you very much, Professor Horwich, for very, very exciting talk. And we will have time for a few questions. And if you raise your hand, microphones will be passed to you.
Hi there. Just a question about the double ring structure. Why do you think that's favored in evolution? Like, why is this double ring structure?
That's really an excellent question. There are actually contexts where single rings seem to be able to be functional. So for example, if you look at liver mitochondria, for example, they have a single ring HSP60 that seems to be able to be quite functional. The part of its cycle that's interesting is the sort of coming around the cycle to the point of polypeptide release. So it has to be able to sort of hydrolyze ATP and allow polypeptide to release without using a trans ring, but it seems to be able to do so. So it is one of the few exceptions of a single ring version of a chaperonin. And it's somewhat open to question whether it is always a single ring or whether it actually could be a double ring at some part during its reaction cycle. But there have been some fairly strong experiments carried out that would suggest that maybe it can stay a single ring through its whole cycle. But in the other areas of nature, these seem to be double rings. So something about ring-ring cooperativity, I'm not sure. Maybe it enhances overall efficiency. I mean, those kinds of experiments are now doable. For example, a Weizmann Institute group has actually measured the ability to change the activity of a particular protein by mutating GRO-EL and watching things evolve in a test tube, more or less. So you could ask questions about efficiency of cellular growth relative to whether you use a single ring version of a chaperonin versus a double, and that would be an interesting question at this point.
So at the end, you show a movie of how the chaperonin works. So it looks like after the protein has been loaded into the chamber, there are not much space between the protein and the chamber itself. So maybe you have a few layers of water there. So my question will be, is there a way you can detect how different the behavior of those water from the bulk water? Or is there some...
Right. So this is a really exciting and interesting question that we have taken up with a lot of people who are in the business of trying to look at solvent versus bound water. So I should say, first of all, at the level of our X-ray structures, none of them are at high enough resolution to start building water in. The best we ever do is around 2.5 angstroms. So, you know, you really have to do better than that if you want to start to build ordered water in. So we've failed as far as that goes, no question about it. With respect to overall water reaching the chamber, there are side holes that are very accessible. So we think there's lots of solvent going in through those side holes all the time. Now the question is, okay, can you put a substrate in there that's so big that you just have 1 or 2 layers of water? I don't honestly know. I mean, one of the phage proteins that's been studied at the EM level by Helen Sable is really huge and seems to just, you know, at the end of folding, it's sort of folded up like an unborn baby inside of the womb. Whether that thing is occupying so much space that you're really down to just a few layers of water, I don't know. Again, I think so far we really lack the tools for being able to sort of look at water right at the apical face. So I've had a number of discussions with Ahmed Ziwail, who's really done a lot of fluorescence kinds of experiments using tryptophan substitutions to look at water at that surface. He says, don't even try. It's going to be way too complicated to do this. So
Professor Ziwail has done some fast the laser trigger experiment, look at the water dynamics next to the protein magneting thing.
There are people trying dynamics experiments, and we've thought about it, but we haven't really tried anything so far. I mean, I think modeling experiments are fine, but ultimately we really do need to do a real experiment, but I think it's pretty tricky to do the O18 kinds of exchanges. Because the problem is you do the exchange, and now you have to rescue fast enough that you don't have back exchange, and the side holes don't go away. I mean, maybe you could go to, to a helium temperature or something to sort of quench the reaction. But then how do you really collect your data? It's an interesting question.
All right, in the interest of time, we have one more question from Ben.
So how is this machine interfaced with membrane for membrane protein folding?
So there has been a bit of work suggesting that GrowEL can interact with some of the smaller membrane proteins. And actually, for example, LAC-Y, it was Gershovich and coworkers and a number of other groups suggested that it could actually be bound in the open ring of the assembly and then discharged in such a way that it would be productively inserted into a membrane. Some of that data has really just sat there without really a further follow-up at a finer level of resolution. So I would say it's a somewhat open question whether GrowEL can act on it. I mean, in principle, it should see anything that exposes hydrophobic surfaces, right? So if you have transmembrane domains that have lots of hydrophobicity, they ultimately should, if the protein collapses sufficiently, it should get into the central cavity, and there should be some interaction with the cavity walls. But whether you really get a productive release, in that context that allows proper membrane insertion or interaction with SEC machinery, for example, I really don't know. So probably more work needs to be done in that area. But it is conceivable.
All right. Thank you very much, Professor Horwich, again.